So, good afternoon, everyone. My name is Chaeyoung Kim, and I'm a master's student in the Department of Experimental Medicine in UBC. I'm also in the RU lab, and I'm also in the registration desk in this conference. So you all have my name tags. And today I'll be talking about my project in UBC, which has to do with decoding airway epithelial responses to wood smoke and air allergen. Okay, so to start off and to make sure that we're all awake, I'm going to start with this interesting chart. This has several different data all together from different sources. And so how many people die from air pollution each year? So globally, it's about 6.7 million to 8.7 million deaths each year is attributed to air pollution, which is a massive global health burden, as you can see. And my project is dealing with one of the portion of that air pollution. Okay, so this is my project, and basically climate change is changing what we breathe. We all know this, but we don't really know the details. So wildfire smoke exposure is increasing from climate change. We all know that. Warmer temperatures and elevated CO2 is also caused by climate change. But we don't know, well, some people don't know, is that air allergen burden is also changing because of climate change. And combined exposure of both wildfire smoke and air allergen may worsen respiratory disease. So, for some background, this is a 2021 study. It's a global time series study, so basically it's from 749 cities across 43 countries, and it's a span across from 2000 to 2016. And it shows us the mortality risk that is related to wildfire smoke. So basically, the big number, 749 cities and 43 countries, shows us that this data is not just from one specific region, but it's a global phenomenon. So what did the researchers find? So basically, I know there's a graph, but I'll explain. Each 10 microgram per cubic meter increase in wildfire PM2.5 is linked to increased mortality risk. So 1.9% all-cause, 1.7% is cardiovascular mortality, and 1.9% for respiratory mortality, which is an estimated 3,503 respiratory deaths attributable to acute wildfire PM2.5 exposure annually. So if we consider all together, it's about 0.6%. So respiratory mortality contributes about 0.6% to all death, including all mortality. But you may think that 0.6% is really small. But when you think about the whole scale, which is entire death, it's actually a very huge number. So maybe I'll just go a little to this chart. So it's relative risk versus wildfire-related PM2.5 concentration. And as the PM2.5 concentration increases, you can see that there's a rapid curve that's increasing, which shows us that as PM2.5 concentration increases, there's actually an acceleration of the relative risk. Okay, so that's about wildfire, and now this is about air allergen. So in our lab, we investigate ragweed pollen, and you might think, oh, what's ragweed pollen has to do with any mortality? Like it's just air allergen, you know? Like you just sneeze a little and that's all. But actually, ragweed pollen increases respiratory mortality. So this is actually from last year in Michigan, and this is for older adults. And high ragweed exposure is linked to increased odds of chronic respiratory mortality. And if you can see where I bolded or like highlighted, the strongest associations are chronic respiratory and COPD related mortality. So at a 7-day lag, if you see the number, it says 2.16 and 2.07 on chronic and COPD. And you can see that the number doubled, which means that double the risk of mortality. And you can see that there's no significant association with infectious respiratory mortality, which actually shows us that the real risk is with the chronic and COPD. So this shows us that there's solid evidence on both sides. So both wildfire smoke and both ragweed pollen shows increase in respiratory mortality, and that is what our lab is here to do. Okay, so that brings us to our aim. So our goal is to characterize how airway epithelial cells respond to wood smoke and ragweed pollen. And we have two hypotheses. Well, I have two hypotheses. Wood smoke and ragweed produce distinct respiratory responses, which means that they will have distinct responses to aerologen and wood smoke, and also co-exposure induced synergistic gene expression changes in human bronchial epithelial cells. This means that so both, they'll be additive for sure, but this is showing us that there's actually like a whole different response when they're added together. Okay, so for people who are not really knowledgeable in airway epithelium, so why focus on the airway epithelium? Well, the airway epithelium is the first place that pollutants and allergens is contact with, and it's both a physical barrier and it's a very active immune signaling tissue. So that is why we use those in identifying biomarkers. Okay, so here is our experimental pipeline. So we start with primary human lung epithelial cells of N equal 40, and then we culture those cells and put them on air-liquid interface, which means that there are growth media on the bottom, but cells on the top, and let them differentiate like a real bronchial airway would do. And then we expose them to wood smoke and ragweed, both distinctly and also in combination. And then we do a lot of readouts. For example, ELISA does screen ILAtes. TEER, which means transepithelial electrical resistance, which has to do with barrier integrity, and RT-qPCR to let us look at gene expression changes. And yeah. So to start with what cells we're using human bronchial epithelial cells collected and banked during research bronchoscopies. So there's actually a few conditions with the respiratory conditions, which are lung cancer, COPD, or asthma, and those are like N equal 30. And we also have healthy controls with N equal 10. So that's 40 donors total. Okay, so before we actually expose them to wood smoke and aerologen, we actually have to establish a primary airway epithelial cell model. So basically, human bronchial epithelial cells are expanded in submerged culture before ALA differentiation. So day 3, day 5, day 15. You can see that day 15, they're very dignified, and you can see it's very concentrated. And so those are basically submerged T25 and T75, and that is what it looks like in an actual ALI culture where we try to mimic the airway. Yeah. Okay, and then when we make all those cells, we put them in the Cultex system. So this is actually a very cool machine. And wood smoke is generated from dried wood in Canada. And it's under controlled conditions in the air pollution exposure lab. So they put wood like here, and then this heater thing goes on and try to burn it in a very controlled way. And then differentiated human bronchial epithelial cells are exposed. So in the left, you can see that they're exposed in the air-liquid interface. So it looks like a little screen, but like these are cells in there, and they're exposed to these tubes. Okay, so we're still going on in this experiment. So these are, Okay, so preliminary results. Thanks to Charlotte Lee and Jennifer Lin. So IL-8 increases with increasing allergen dose. So no significant wildfire smoke effect at control or low ragweed dose, but we did see a huge increase at highest ragweed dose to 1 to 10 dilution. So these are allergen concentrations. And you can see wood smoke versus non-wood smoke. So with wood smoke, there's an increase. Okay, so TEER is basically barrier integrity. So we put the TEER machine onto the cells and try to check how the barrier integrity basically. And then so wood smoke increases here at control and 1 to 10 dilution dose. And there's a positive but no non-significant trend at mid-allergen dose at 1 to 20 dilution. So this shows us that they're sensitive enough to detect what's going to happen later on and in the full study, giving us confidence. So basically, what's next? So we haven't done this yet, but we will, hopefully soon. So RNA-seq and bioinformatics analysis to actually, so remember when I talked about we're going to characterize the responses? So this is what's going to happen. So we're going to collect RNA for RNA-seq sequencing to profile genome-wide epithelial responses. Yeah, I'll let you read them. Okay, so I'd like to thank my supervisors for giving me the opportunity. And also I'd like to thank Charlotte Lee and Jennifer Lim for those awesome preliminary results. And I also want to thank Genome British Columbia, Center for Lung Health, and Vancouver Coastal Health Research Institute. Thank you.
Thank you for your presentation. You mentioned that the samples of the bronchi cells were collected from those with lung cancer, COPD, and asthma. So I have two questions related to that. I was wondering if you saw any differences among different conditions. And for asthma, I believe that they're reversible, so like their condition worsens at certain situations, and that's when they have like that observation. I was wondering if that impacted your cell collection.
So we're trying to minimize different variables in this project, and I'm sorry to say that we're still in the very preliminary part. So we haven't really sequenced any of the RNA that we've collected, but so far when we just look at those tier data, when I'm like tiering, so variant integrity, I don't see that much of a difference yet. But the real key is in the DNA RNA that we'll be using transcriptomic and also bioinformatics. So we'll let you update it.
Thank you.
Okay.
And so I've heard that pollutants become more concentrated like in northern communities. So I was wondering if you've like looked into whether northern communities and indigenous communities are at exacerbated risk.
Okay, so we do have a very diverse pool of participants from the bronchos. Well, I think you're talking about this one, maybe? Okay, so we do have a very diverse, so we will be actually doing, we'll be using different, like, if they're like sex, basically a woman, man, like female, male, age, and basically like different backgrounds, ethnicity, and yeah, so we'll be considering all of that. Yeah.
Hi, Kim. Thank you for the great presentation. I just wanted to ask how you came to hypothesize that wood fire and wood smoke and aeroallergen would have synergistic effect, because at first sight these two don't look like they're related. They're not related. So yeah.
So the most important thing is actually I have forgotten to anyway, but like literature, like right now, they've only experimented with only wood smoke or only aeroallergen or only like maybe PM2.5 like Korea and 미세먼지. But like the most important thing is that when we're here in Vancouver, we're not just standing in front of the tree and smelling all the wood smoke that's coming from directly. That is passing through all the aeroallergen, sunlight, like UV light, and it's very aged because it takes several times to reach you in Vancouver or maybe Montreal. So that's why we wanted to see if aeroallergen, especially with increasing climate change and increasing aeroallergen exposure and increased mortality, if that's going to make a huge difference in especially vulnerable patients with COPD and asthma. Yeah.
I hope you find this interesting. Thanks.
Thank you so much.
Thank you so much.